Hi, everyone. This is Roberta Hansman. I'm the staff chemist at the National Ocean Sciences Accelerator Mass Spectrometry Facility, or NOSAMS, and we are located at the Woods Hole Oceanographic Institution on Cape Cod in Massachusetts. I want to thank the organizers of this radiocarbon workshop for the opportunity to give this presentation, and I'm going to talk about external laboratory submissions, analysis, and data reporting of samples for radiocarbon. I want to start with a brief overview of the NOSAMS facility, which was established at Hui in 1989 through a cooperative agreement and with continued support from the U.S. National Science Foundation. In the 30+ years since, we have reported more than 150,000 radiocarbon measurements. And in the past several years, that has looked like an average of 6,400 analyses of unknown samples annually. And those unknowns include many different sample types. Some of the most common ones are indicated here, both inorganic and organic carbon, carbon dioxide gas, carbon dissolved in water, both inorganic or DIC and organic or DOC. This is a rough breakdown of the average proportions of those sample types each year. The facility was created to analyze radiocarbon in seawater DIC, through which we've learned a lot about ocean circulation and ventilation. And we continue to make 1,000 seawater DIC measurements each year, collected through the Global Repeat Hydrography Program. So those are included in this large slice here. In addition to those 6,400 unknown sample measurements, we also process and analyze 3,600 standards and blanks for all of these sample types. So in a given year, we're making around 10,000 radiocarbon measurements. These photos at the bottom are from the facility. This is our sample preparation lab showing many of our different vacuum lines and some of our staff. This is, of course, in the before times, as no one is wearing masks in the facility in this photo. And this is one of our AMS systems shown here in the bottom. And this is Mark Roberts, pictured here, our staff physicist. For this presentation, I broke down the general process from an outside client preparing and submitting samples to us for radiocarbon measurements, to our processing and analysis of those samples, and then the reporting of the data. So I'm going to go through these steps briefly and give some hints and things to think about if you're submitting samples to a facility for radiocarbon analyses, as well as some details about what we do with those samples and the data we generate. The first step of the process is for you or the outside client to prepare your samples. It's important to define your samples. And by this, we mean both making sure they are clean and free from any debris from collection or sampling that is not part of the material that you're interested in dating. And also, if the sample is large, be sure to subsample as needed, just as you might isolate or pick specific forams for a large shell or piece of wood. For example, you should clearly identify and subsample which part of these larger items you want analyzed. And as you're doing this, and even before when you're selecting samples, keep in mind sample sizes. At NOSAMS, our target mass for radiocarbon analysis is 1 milligram of carbon. For an inorganic calcium carbonate sample, these are around 10% carbon, so you want to shoot for 10 milligrams or so of sample mass. For organic samples like plant material or wood, these are 40% carbon. Those samples for bulk analysis, such as sediments, these materials can have a wide range of carbon content down to a few percent or even less. We can and frequently do make robust measurements on small samples, which we define as 25 to 100 micrograms of carbon. But it's important to keep in mind that as sample size decreases in this range, the precision of the measurement worsens, or the errors increase, to put it another way. So more material is usually better to a point, though we certainly understand that sometimes your samples can be very limited in size. And finally, you should also minimize contamination of carbon that can be added to your sample as you collect and prepare. It's important to wear gloves so as to avoid transferring carbon or oils from your skin to the sample, and samples should be dry to prevent microbial growth. And most critically, here, you must avoid contamination from elevated unnatural tracer levels of C14. Hot samples can compromise your own science, but also significantly impact our labs, as we have to thoroughly clean and replace anything that the sample came in contact with. It also can have an impact on the samples from other clients that may have been processed at the same time as your hot samples. So we must be careful to protect our facility, and all, I imagine, natural abundance radiocarbon facilities are struggling to always protect their facilities from contamination. So please take the time to investigate the history of the lab spaces and equipment where you collect and prepare samples before submitting them. And if you suspect tracer work, the first thing we would recommend that you do a swab test to test for gross contamination. And then we also can perform swipe tests to really dial down and make sure that you don't have tracer level contamination in your lab space or with your laboratory equipment. When your samples are prepared, they'll need to be logged into our online submission portal, and it's located at this website, and you'll log in. And then the system is pretty straightforward. So just briefly, you can select here to submit samples. And after clicking through and answering some questions, each sample can be entered into this spreadsheet online. And we need information about Sample type, this is a drop-down menu where you can indicate if you have plant or wood, charcoal, bone, coral, etc. There's places here to add sample mass, percent organic carbon by weight, if you know that. You'll also need to select if you would like to have a split of your sample measured for delta C13 by IRMS. And if your samples need any special handling or processing, this should be indicated in the submission. And after submitting your sample info on this sheet, as you click through, you'll then provide funding information as part of the submission. Once the samples have been submitted into the portal, they can be shipped to us at NOSAMS. It's important to use clean glass or plastic vials and think about things like the size of the opening of the vial. If it's narrow and it was difficult for you to get your sample in, it's going to be difficult for us to also get it out on our end. And be sure to label them clearly and secure them well for shipping. When we receive the samples, we will send you an acknowledgement email and look them over. If we have any questions, whether with labeling or any changes or specifics for how we should process them, this is the time where we will start to contact you and begin any kind of dialogue that's needed as we begin to work with your samples. After you receive your samples, check them over and log them in. The first step of our sample processing is to convert the carbon in the sample to carbon dioxide or CO2. And that is done a little differently for different sample types, but briefly for inorganic or carbonate samples, such as the pieces of coral shown here, we acidify these samples with phosphoric acid, which converts the carbonate to CO2. We use these specially designed vessels where we weigh out the sample in the bottom here. And we add acid to the arm. We evacuate this on a vacuum line and then tip the vessel so that the acid is added to the sample and generates CO2, and the generated CO2 is contained within this vessel. Organic samples are typically first pre-treated before converting to CO2. This usually involves a series of acid and base rinses. This removes both inorganic carbon as well as any base-soluble organic acids in the sample. Following pre-treatment, we combust organic samples to CO2 in the presence of oxygen. This is often done using an elemental analyzer, which is shown here. We can also do this in sealed quartz tubes with copper oxide. For water samples, DIC is also acidified to generate CO2, which is then sparged from the sample, as shown in the photo here, or extracted over a membrane on a system which we have here that we call Redix. Organic carbon dissolved in water is oxidized to CO2 using UV light. These are two separate samples in reaction vessels inside a chamber with a UV light inside, and the UV light is used to oxidize the sample, the DOC. It's converted to CO2. This is that chamber on a vacuum line, and then the CO2 that's generated is purified and quantified on this line. Once we have the CO2 from all these different sample types, the next step is to reduce the CO2 to filamentous carbon, which we refer to as graphite. This is done with hydrogen and heat on an iron catalyst by this reaction shown here, and we remove this water with magnesium perchlorate. We have several automated graphitization lines in the lab. The one pictured here is our newest one. But basically, to look at this in a little more detail, this is a reactor with the iron catalyst where the CO2 sample will be transferred to. First on the system, it is purified to remove water and any non-condensable gas and quantified. Hydrogen is added, and then these insulated ovens are placed over the reactor for the reduction to proceed. This line has 10 reactors, as you can see. And then after the reduction, what you're left with is these tubes of graphite that are formed on the iron catalyst as shown here. We analyze that graphite using Accelerator mass spectrometry, or AMS. The graphite is pressed into these sample holders that are mounted on a wheel and then loaded into the source, which is pictured here. The wheel has 134 positions, and alongside the unknown samples will be several primary standards interspersed throughout that are used to normalize the data, as well as other secondary standards with known radiocarbon values and blanks, or material that is free of radiocarbon or radiocarbon dead. This is a schematic of our CFAMS accelerator with the source shown here. The samples are sputtered with cesium to generate negative ions. These are extracted and accelerated through the system. And we detect C14 ions while simultaneously measuring C12 and C13 ions in Faraday cups. So ratios of C14 and C13 to C12 are determined on the system. The C14 to C12 ratios we get from the AMS system are corrected for fractionation using the AMS C13 to C12 ratios. And they're also normalized to our primary standards, which are OX1. These are modern standards, and so we're determining the fraction modern, or the deviation of a sample's radiocarbon content from this modern standard. This plot here is showing the normalized ratio of an OX1 standard measured 10 times, as we typically do for each sample, standard and blank. The fraction modern is an average of these 10 measurements and the accompanying standard error. We then correct the data for all samples using an appropriate blank for the sample type and the processing that occurred during the preparation of the samples from CO2 and graphite. And we propagate these errors for these corrections. And so when we report data to outside clients, we report this blank corrected fraction modern, or FM, and the propagated error. When we report results, we send a spreadsheet by email, which can also be accessed through our client portal. In addition to fraction modern, we calculate radiocarbon age using this equation. These ages are reported without reservoir corrections or calibration to calendar years. And if a sample collection date is specified, when the sample is submitted, we can also report big delta C14, which accounts for the decay between the time of collection and the time of measurement. So that's it. It's the whole process from preparing samples to submission to our reporting of data. If you have any questions, feel free to contact me at this email address. And you can also have a look at the NOSAMS website, where we have lots of resources and more detailed information about several of these steps. Thank you.